Bismillahirrahmanirrahim. We start another paper 3, which is a practical exam paper, which is going to be for 2 hours, and we're going to trying to discuss the question 1, so everybody can do this more befittingly and can do this paper, even though you have not had much lab practice, but still you can do this paper without a lot of lab practice as well. This paper is for 2 hours. And you have to do question 1 in 1 hour, and the second question in another hour, and one is a microscope question, and one is always a practical question. So remember this, 2 hours, and you could be either doing the 3-3 or you could be doing the 3-4. So in Pakistan we have 3-3 and 3-4. In other countries, of course, we have the other variants. Now we start with question number 1. Now you read the question very carefully and you underline the keywords and you read the question at least twice. You are provided with a solution labeled E containing an enzyme. So E for enzyme, E is the labeled solution E. So there would be a sort of a solution here, and on the beaker would be written E. And this instruction would come to us from Cambridge when we were going to set up the practical in whichever school you're going to give the exam. So you're provided with a solution E containing an enzyme. So there is some sort of fluid in it, and that fluid contains an enzyme which coagulates milk. Now, enzyme E hydrolyzes, breaks peptide bonds between certain amino acids in a protein found in milk, and this results in the coagulation of milk. It means the clotting of milk. Calcium ions are needed for this coagulation, right? So we read the question and we understand the question, and then we look at this word coagulation and we try to explain it to ourselves. Some of you don't really understand the word coagulation, so I usually say use the word clotting, like blood clots. So if you don't understand the word coagulation, try to understand what it means. Clots milk, here it says also clots milk. When a mixture of milk, calcium chloride solution and E, so we have milk, then we have the calcium chloride, and we have the E, is gently turned in a test tube, the milk will coagulate, producing lumps of a white solid in a liquid. And the end point of the enzyme catalyzed coagulation is when all the milk coagulates as shown in figure 1.1. Now you can see this picture which I have brought in, and I want you to see that how this clotting is going to look like. It's going to be something like this, which I have brought in this picture. This wasn't in the paper, but I have just brought this in to show it to you that this is what the coagulation would look like. So you can see there's a liquid, there's a watery part, and then there is the white solid, which is the coagulated milk. Just like you see it at home in yogurt, that's also coagulated, or if you have seen, when the milk goes sour, then it coagulates. You will investigate the effect of temperature on the time taken to reach the end point, which means the time taken for it to coagulate. You are provided with materials as shown in table 1.1. M is the milk, M for milk. It's 100% milk, there's no hazard, and it's 150 cm cube given to you. E is the enzyme solution. It's a harmful irritant, and there's only 20 cm cube given to you. And C is the calcium chloride, which is a harmful irritant, and it is 20 cm cube given to you. Of course, all this instruction comes from Cambridge, and whichever place is your center, they will have all this information beforehand as to provide you with all this. If C or E comes into contact with your skin, wash off immediately under cold water. It is recommended that you wear suitable eye protection. You will test the activity of an enzyme E at 30 degrees Celsius and other temperatures up to a maximum of 50 degrees Celsius. So they're giving you all this information. You need to read all this information and you need to process all this information in your mind that, okay, what are we going to do? Now it says A1. Complete table 1.2 to show three other temperatures that you will use to show the effect of temperature on the activity of enzyme E. And this for 1 mark, and they've shown you 30 degrees Celsius and 50 degrees Celsius. You see how they've written the units in the heading, and then you have to give me any other 3 different temperatures. Now as you can see, I have written 35, 40 and 45 because between 30 and 50 you have it evenly spaced, and that will give you a better this thing. You can't say 37 and 42 and then 48 or something like that. So they must be evenly spaced, and that's how you do it always. That's a rule that we follow. Now we read the rest of the paper, and it says carry out step 1 to step 14. Now let's start doing it. I'll try to do a little diagram to show you. I've set up a water bath ready for step 6. The starting temperature of the water bath should be 30 degrees Celsius as shown in table 1.2. So you set up a water bath, and you've got a thermometer, so you're going to measure the temperature, and they give you hot water and cold water, and you keep it at 30 degrees Celsius. Water bath means it has a beaker with water in it. Now in another test tube, put 10 centimeter of M. So in this we have M, and that is 10 centimeter cube. So you're going to use a syringe and put 10 centimeter of M into a test tube. Repeat step 2 so that you have 2 test tubes of M. So we have 2 test tubes with M in it, 10 centimeter cube, right? So repeat step 2 so that you have 2 test tubes containing M. So this is one test tube and this is the second test tube, and you have now M in both the test tubes. Put 1 centimeter of C. So you put 1 centimeter of C, so 1 centimeter cube of C. C was calcium chloride. And you do the same in this. So put 1 centimeter of C into each test tube. So you put C here, you put C here. Gently shake the test tube. Now gently shake means, of course, this is a very error prone. Why? Because gently shake, for somebody it might be just shaking it slightly, somebody it might be doing it more severely because he or she is very angry and something bad happened to them in the morning, they didn't have their breakfast and their mom shouted at them, so they're in an angry mood, so probably they'll shake it a little harder. But it's a very it's actually there are mechanical stirrers as well, and they're turned on at a certain speed. So gently shake the test tube to mix M and C. So error is in the mixing. You should have a mechanical stirrer to mix it. Put the test tubes into the water bath and leave for 3 minutes. So then you put these in the water bath. Put both these test tubes in the water bath, right? So this one comes here and this one comes here. And leave it for 3 minutes. And of course, you can be 3.5 minutes or 4 minutes. You can probably it's not going to be exactly 3 minutes. I know that. Now it says explain why the test tubes are left in the water bath for 3 minutes in step 6. You see, you've still not added the enzyme. Why we've done this is the contents of the tube. What were the contents? The contents were M and C. So the contents of the test tube reach the temperature. Contents of the test tube reach the temperature because you want the uh, milk and the calcium chloride both to be at that temperature, which is 30 degrees Celsius, because you're going to add the enzyme and then test it. Now you've left it, so you've left one test tube. Then it says remove one of the test tubes from the water bath. So that means the other one is still there, and that's probably more than 3 minutes. That's going to be then 5 minutes in the water bath. So remove one of the test tubes from the water bath, and now it comes to a very interesting state. The process of coagulation will start when E is added to the test tube. So you've taken this test tube out, and now it's got the M in it and the C in it, right? The C was I made in orange, the M I made in blue. Now put 1 centimeter cube of E. Now you have 1 centimeter cube of E in the syringe, so that it runs down the side of the test tube and forms a layer on the surface of the mixture, so that it goes down here, goes down the side, and then forms a layer on the top, as shown in figure 1.2. So syringe, E in the syringe, test tube held at an angle, E running down the inside of the test tube, mixture, layer of E forming on top of the mixture. So now you've got this E on top of it. Right? I hope everybody's following me. Gently shake the test tube to mix the solution and start timing. So then you shake it. Now you rotate the test tube and continue to rotate it while observing the mixture until the end point is reached. Stop timing when the end point is reached. Maybe it takes 20 seconds, 30 seconds, 60 seconds, 90 seconds. I don't know how much it takes, right? We'll talk about that a little later. If the endpoint has not been reached by 180 seconds, now this gives you a clue that it has to be less than 180 seconds. Stop timing and record this as more than 180. Now record in A3 the time to reach the endpoint. Underline this so that you remember to do this in A3. The time to reach the endpoint. So you know immediately in the table there is something going to be time and seconds. The table heading is going to be time and seconds. Repeat step 7 to step 11 with the other test tube in the water bath. There was another test tube, remember? Remember there was another test tube here. We had two test tubes here. 1 and 2. So we have two test tubes here. So it says then, repeat step 7 to step 11 with the other test tube in the water bath. Then step 13, set up the water bath at the next temperature after 30. So the next temperature was 35 degrees Celsius. Now you set up the water bath at 35 degrees Celsius. Repeat step 2 to step 13 for each temperature stated in table 1.2. So you do it for 35, then you do it for 40, then you do it for 45, and then you do it for 50 degrees Celsius. Right? So you repeat step 2 to step 13 for each temperature stated in table 1.2. Now coming to A part 3, which we mentioned earlier, record your results in an appropriate table. And look at the marks, 5 marks. Now everybody should get their 5 marks in this. There is no way that anybody could lose any marks in this table. So always what you do is, you write the headings first. So the heading was, in the first question was, what did you change? What did you change? You changed the temperature. So when you change the temperature, that is the independent variable. I always say independent I. I decided to take 5 temperatures. I decided to take 5 pH. I decided to take 3 different types of plants. So what did I do? That is always the independent variable, and that is always the first column. That is always the first column. You must write it first, the independent variable. And the second is the time in seconds. This is the dependent variable. What did you measure? You measured the time it took for it to coagulate. So you wrote both this and you got 1 mark. And you didn't do anything else in the table. You didn't complete the table even. But you wrote this, you got 1 mark. Okay, now let's complete the table. You took 30. Please do not write degrees Celsius here again. If you write that, it's a -1. 35, 40, 45, and 50. And now you can draw the lines on it. I always find it easier to first write it and draw the lines because I usually don't get the lines right. But maybe you all are good at it. I'm not so good at the lines and doing this. Now you remember there were 2 test tubes. So let's make a column here again because we had 2 test tubes. Now I've completed the table for you, and you can do this in the exam also. You can do one experiment and then just cook up all the results. It's a very easy way. You have to be just intelligent enough to do that. You do one and you do one on 30, 150. The rest of it, you can just write it, make it up, cook them up yourselves. Now, what were the 5 marks for? Number one was for the heading, 1 mark for this. The second was records times for all the temperatures. So you gave me a record for all the temperatures, whatever the time. We're not talking about less or more. There was only 1 mark that the highest temperature should have had the least time, should have happened very quickly, fastest time. For the highest temperature, the fastest time. So for 50 degrees Celsius, it should have been the least amount of time, fastest time. Then you recorded the repeats because you did it twice. So you repeated it. So this was one time you repeated and the second time you repeated it. And it records to the nearest whole second. Please do not write it in fraction 60.2 seconds. But in fact, the stopwatches that we give you does record it in fractions, but we can't find those other stopwatches that we have here in Pakistan. It does give you in fractions, but please do not write, always write in whole seconds. This is a rule that I tell you all to follow. Whole seconds. Never write 60.2 seconds or 20.4 seconds. Don't write that. Always write in whole seconds. Records to the nearest whole second. So these were the 5 marks. Everybody should have got these 5 marks. Everybody must do these paper threes like a theory paper, like I'm doing with you now. And if you have any problems, you can always message me on my WhatsApp. My number is there. I'll leave my number at the end as well. And you can always WhatsApp me if you have a problem in doing these tables. Now it says part 4, state the independent variable. You know that was temperature. What did you change? You must know what is dependent and independent variable. If you don't know it, just Google it and have a look at it. What is dependent? We do a lot of this in A2 as well. So you must know it. You must be very clear about it. So temperature was the independent variable. Now it says suggest a suitable control for this investigation to show that it is the enzyme E that coagulates the milk. Naturally, you just use boiled and cooled enzyme. Boiled and cooled enzyme. So if you use boiled and cooled enzyme, you know that the enzyme is not going to work if it is boiled. And if still that reaction takes place and still coagulates, then it means it wasn't the enzyme, it was something else. Either you said boiled and cooled enzyme or you said replace the enzyme with water. You can do that, or you can say replace the enzyme with water. Do not use the enzyme. Now be very clear when you write this. Be very clear in the English. Some of you struggle with English. Please improve your English. Replace enzyme with water. Now coming to part 6, the procedure described by step 1 to step 14 investigated the effect of temperature on the activity of enzyme E using the time taken to reach the endpoint. Describe how you would modify the procedure to investigate the effect of changing the concentration of milk on the time taken to reach the endpoint. So the first is from the question you pick up stuff. Now you would, of course, temperature would be kept constant. Now you will not change the temperature. In the previous one we changed the temperature. Temperature is kept constant. Whatever temperature you might choose, 30 degrees Celsius or 40 degrees Celsius. So you would keep it constant at say 35 degrees Celsius. Then you would use 5 concentrations of milk because you're going to change the concentrations of milk. By how will you change the concentration of milk? So you have a 10% solution, you dilute it. So you can either use a serial dilution, either you use a serial dilution to make the different concentrations, or you do something which is known as proportional or simple dilution. So now very simple, the things from the question that you have to pick up. How you would modify the procedure to investigate the effect of changing the concentration. So you keep the temperature constant, 5 concentrations of milk and 3 marks and you get your 3 out of 3. So you modify this procedure. So in this procedure, we change the temperature. So okay, here now we will not change the temperature because we are not studying the effect of temperature. We're studying the effect of concentration. And you use at least 5 concentration. Uses 5 concentrations of milk. And how do you make the concentration is by either serial dilution or by proportional dilution, and which is of course also called simple dilution. And you must know how that is. Of course, they're not asking you the details of it, but you just have to write it. B part of the question, a scientist carried out an investigation with the effect of enzyme concentration on the coagulation of milk. The scientist calculated the activity of the enzyme for each concentration. The results are shown. Now, as I told you before, this is the first column. This is always the independent variable, and this is always plotted on the x-axis. So it becomes easy. The rest of this goes on to the y-axis. You have to write the whole thing, activity of enzyme slash arbitrary units bracket AU, and you have to write the whole thing for this percentage enzyme concentration on the x-axis. Do not write any abbreviations. Do not miss out the units. If you miss out the units, you lose a mark. Part 1. Plot a graph of the data in table 1.3 on the grid in figure 1.3 and use a sharp pencil. Why? Because sharp pencil, you got to put a dot. You don't have to put a blob. You have to put a dot. And this is why you have to be careful with a sharp pencil. Now, so you start labeling the axis. Now you can see here that I have plotted the points, and there's a mark for plotting the points, and then there is a mark for connecting them. So you've got to remember is first you plot them and then you connect them, and that's why you use a sharp pencil. Now the labeling of the axis got you 1 mark. The scale, which is 0.05 to 2 centimeter, and you must label every 2 centimeter. And on the x-axis it was 0.05 to 2 centimeter. On the y it was 20 to 2 centimeter. And this is of course given in the mark scheme. If you didn't do this scale, well, you won't get a mark for that. Correct plotting, and then 5 points joined. So you join the 5 points. Please do not extrapolate. So you join the points, and then you, I'm sorry, it got a little crooked at the end. And you do not extend it to 0 or anything. So you just join the 5 points. Do not extrapolate means do not extend it. So 5 plot joined with a thin line passing through all the points, and line is either smooth curve or joined plot to plot. This is what I'm reading from the mark scheme. And this will get you your 4 out of 4. So everybody should know that how do we going to do this part of the question. Then coming to part 2 of the question, use your graph to determine the activity of the enzyme when the enzyme concentration is 0.25. So this is the point where we have to see it. Show on your graph how you determine this value and activity of the enzyme. So you have to show it on the graph. You can see I've drawn this black line, and you can see the reading is 80 arbitrary units. So all I have to do is, if I didn't draw this line on the graph, and I'm not going to get 1 mark, and there are 2 marks for this, so just writing 80 here would not have got me the mark, because it says correct estimates based on the candidate's graph and shows the intercepts on the graph. So the 2 marks are number 1 for that line on the graph and for the correct answer. Describe the trend in the result shown by your graph. What is the trend? It's a very simple trend. As the enzyme concentration increases, the enzyme activity increases. It's a very, very simple graph. You can see as the enzyme concentration is increasing from 0.05, 0.10, 0.20, 0.25, the activity of the enzyme is also increasing. From 2 it went up to 4, 5, 6, 7, it's increasing up to 20, 40, 60, 80, so it's going to increasing. So you just had to, that's a very simple answer. I'm surprised if any student doesn't get that answer. So you could have easily lost this mark if you just wrote it very casually that enzyme activity increases. No, you have to write the whole thing. As enzyme concentration increases, the enzyme activity increases, and that got you your 1 out of 1. Last part, suggest an explanation for the trend. Why is the enzyme activity increasing as the enzyme concentration is increasing? What does enzyme concentration means? More enzymes present. If we say 10%, then we say 40% enzyme. So that means 10 active site means 40 active sites. So more enzymes present. When the concentration increases means more enzymes present, which means more active sites. There is just one enzyme or there are just two enzymes. And now there are 6 enzymes. So there are 6 active sites. There were only 2 active sites previously. So more enzymes present, more active sites. That is the right wording. You can say more enzymes. That's not going to get you any marks. And that means more enzyme substrate complexes. And you can abbreviate that. ESCs means enzyme substrate complex. So very simple, 2 marks. More enzymes present means more active sites, more enzymes present, so more enzyme substrate complexes forms, more product formed per unit time, so the activity increases. So these are the exact words that I would like you to use. More active sites, more enzyme substrate complexes formed, or you can have said more successful collisions. That was, of course, if we had temperature coming in here, more temperature, more kinetic energy, more successful collisions, more products formed per unit time. So the rate of product formation would be faster, which means the enzyme activity is faster, because the rate of product formation tells you the rate of enzyme activity. I said the rate of product formation. So thank you very much, and this completes question 1. We will continue with the other question in the next video.